Very good evening. Today we discuss on one is the process, central dogma, which includes three steps. That is replication, transcription, at last translation. Here DNA forms DNA again what we call is replication. DNA forms RNA what we call is transcription. RNA forms proteins what we call is translation, and RNA forms DNA what we call is reverse transcription, which takes place in the retroviruses. Today we are in the discussion of transcription, how it takes place. Will you have a look at this? Here transcription, what is this transcription is formation of mRNA from the DNA template, what we call is transcription. Here the DNA template at the transcription level, it has three parts: promoter and the structural gene and a terminator. Here the promoter region, as I indicated here, the double stranded DNA, this is the transcription unit, the double stranded DNA, which act as a template for the synthesis of mRNA. Here this mRNA, it is a very rich with the nitrogenous bases like A, T, A, T, A. If you read this, it is like a T, A, Tata box. So the promoter region of template DNA, which undergoes the transcription, is very rich with the Tata box. Also what we call this Tata box is the Pribno box, we call Pribno box. And here transcription always takes place from the 5 end to the 3 end. What is this 5 end and 3 end? We already discussed in the structure of DNA. Here if you see the elongation of mRNA on DNA template strand takes place with the help of an enzyme called DNA polymerase. The DNA polymerase, it is the enzyme very rich in nucleoplasm of eukaryotes and prokaryotes, and this DNA polymerase consists of two parts. That is, the DNA polymerase, actually it is the holoenzyme, and it consists of a core enzyme and a sigma factor. Here, what the purpose of this sigma factor is? This sigma factor, it recognizes the starting signal of promoter. Wherever the promoter region is present in the double stranded DNA, that can be recognized by this sigma factor. This sigma factor, it directs the binding site of double stranded DNA to the RNA polymerase into the promoter region. And here this RNA polymerases in eukaryotes are of three types. We'll see here. Here in eukaryotes, three types of RNA polymerases. This RNA polymerase is specialized to synthesize special type of or different type of or particular type of mRNAs or different type of RNAs. Here RNA polymerase 1, if you see, it is a precursor of rRNA. RNA polymerase 2, it is a precursor of snRNA, small nuclear RNA, and mRNA. RNA polymerase 3 is a precursor of tRNA and rRNA. See here, the process of transcription, it goes simply in the eukaryotes. If you see the prokaryotes like bacteria, This transcription takes place in three steps, that is initiation, elongation and termination. If you see the initiation, this RNA polymerase, which consists of a sigma factor, it binds with a promoter region, the promoter region which is very rich with the Tata box, as I told here. And here, after the initiation process, immediately the process elongation starts. What is this elongation? Is elongation of mRNA strand. Here, if you consider this, all the area is the cell or the nucleus. The nucleoplasm is very rich with a lot of nitrogenous bases. This nitrogenous basis comes inside the RNA polymerase in a complementary coding manner, and it attach each other. All the complementary codes, they attach each other, they sense RNA strands out. And here, when this RNA polymerase binds with this promoter region, this RNA polymerase temporarily it unwinds the double-stranded DNA into single-stranded RNA temporarily within this DNA, whereas the DNA is very tensed to helix each other. That's why once this RNA polymerase moves to the next helices, immediately it binds together or it helices together. That is what the double helical structure it is. So this double helical structure will not be hampered. This double helical structure will not be disturbed by this RNA polymerase. Why? Because this RNA polymerase is acting temporarily to make the synthesis of mRNA. Then after, what will happen is this elongation completes. When it completes is when this RNA polymerase reaches the site of the terminator. Here the terminator region will be. When it reaches the terminator, immediately this RNA polymerase comes out. So the RNA polymerase and the mRNA strand will be detached from the double-stranded template DNA. Then after, what will happen is the newly formed this mRNA, how it looks is here. In a eukaryotes, the primary transcript, that is what mRNA, it consists of two parts, exons and introns. Exons, where they express themselves, they are functional units we call. Introns are non-functional units, they are called as interrupting sequences. If you see, this is what the newly synthesized mRNA, where here this newly synthesized mRNA is here now. This newly synthesized mRNA consists of exons and introns. Here the exons are expressing sequences, introns are non-functional or interrupting sequences. Here these introns should be removed from the exons sequence, then the mRNA will be the functional completely, and the transcription process also will be completed by the termination of introns by special type of enzymes, what we call is spliceosomes. These spliceosomes cuts introns and makes the exons join. Then this type of complete mRNA takes place, which is a functional. By this, almost all the transcription process complete. The process of termination of introns from the exons, what we call is the splicing process. After the splicing process, to conclude this transcription process, this primary transcript should undergo, one is a process what we call is the capping and tailing. What is this capping is? Methyl guanosine triphosphate should be added to the phi end. Here methyl guanosine triphosphate should be added to the phi end, and at the 3 end adenylate residue should be added, so that the complete structure of mRNA forms here. By this, transcription process will be completed, and we'll have a overall look on this transcription process in a small video form. Here RNA polymerase. This is what it consists of: initiation, elongation and termination. Here the promoter region which is present on the double stranded DNA, this RNA polymerase binds. Once it binds to the double stranded DNA, temporarily it unwinds the double stranded DNA internally as such. And here nitrogenous bases in the complementary sequence, like a complementary base pairs, like it attaches and will be terminated from the immediately from the double stranded DNA, whereas double stranded DNA it is going to bind or helices each other again. The mRNA comes out. Once this RNA polymerase reaches terminator region, immediately the newly synthesized mRNA and RNA polymerase detaches from the double stranded RNA. Then after, immediately this mRNA consists of, as I said, exons and introns. Here the exons are the functional units, introns are the interrupting units, will be separated by an enzyme called spliceosomes. The spliceosomes comes together here. Spliceosomes comes and they remove introns to join the exons. By this, the complete formation of mRNA taken place, and capping and tailing also we can see very clearly here. So by this we are completed the process of transcription. Thank you.